Every morning, millions of people swish a harsh, bright blue liquid around their mouths, hoping to obliterate every single microscopic organism hiding between their teeth. We treat the mouth like a contaminated surface that needs to be sterilized. However, aiming for a perfectly germ-free environment actively sabotages human biology. Based on findings from the Human Microbiome Project, this data visualization plots markers for oral pathogens in two groups of people. On the left are profiles of individuals with oral disease, and on the right, perfectly healthy mouths. Notice how the pathogen markers are heavily present in both columns. The data shows that the mere presence of bad bacteria does not dictate a disease state. The healthy group's ecosystem remains perfectly stable, even while hosting the exact same pathogens. If the very organisms we blame for cavities are happily living in people with perfect teeth, then carpet bombing the entire oral ecosystem is an ineffective approach to disease prevention. Since having pathogens in your mouth doesn't inherently cause decay, we have to look at what actually triggers the damage. Disease occurs when specific acid-producing bacteria, such as bifidobacteria and lactobacilli, overgrow and disrupt the local environment. Later research confirmed that the exact mix of microbes driving those cavities varies wildly from person to person. Your mouth is a highly individualized ecosystem. Managing it requires targeted adjustments based on your specific biology, rather than a broad, scorched earth chemical attack. A healthy mouth exists in a state of homeostasis, where a diverse community of microbes works together to provide vital services for the human host. For example, the resident Streptococcus salivarius actively works to inhibit inflammation along the gum line. These microscopic residents also operate a chemical processing plant that services the entire cardiovascular system. Chewing fruits and vegetables releases natural nitrates. Oral microbes immediately latch onto these molecules, transforming them into nitrite. Once swallowed, your body converts this nitrite into nitric oxide. This specific molecule signals your blood vessels to relax and expand, directly regulating your blood pressure. This transformation links the status of the oral microbiome to the body's mechanical regulation of blood pressure. Which brings us back to the widespread use of over-the-counter antiseptic mouthwashes. These products act as a broad-spectrum wipeout. They destroy the beneficial oral microbes alongside the pathogens. When you destroy that bacterial workforce, you directly interfere with the nitrate-converting process. Using antiseptics to protect the teeth inadvertently sabotages the body's natural blood pressure regulation, creating a need for a more targeted solution. The challenge is finding a way to reduce plaque and disease without the collateral damage of wiping out our microbial helpers. In 2024, researchers led by Divya Bharathi published a clinical study testing an alternative approach, targeted herbal mouthwashes. This chart compares a chemical antiseptic, chlorhexidine, against a botanical blend. For plaque and bleeding reduction, the bars rise together, matching efficacy. But when measuring gingival inflammation, the botanical blend distinctly outperformed the chemical alternative. Because these specific botanicals minimize side effects and preserve the microbiome, they provide a scientifically supported alternative to harsh chemical agents. Using this research, we can employ oral health protocols engineered to curate rather than clear-cut the oral environment. An effective protocol relies on a precise combination of researched ingredients: cinnamon, raw bee propolis, curcumin, piperine, triphala, and clove. Maintaining biological homeostasis through targeted botanicals provides a path to oral health that respects the body's natural systems.